Hi, Professor and my classmate. I'm Yuhan from Gene Expression Research Lab under the supervisor, Professor Hong Jong-chan. Today, I will introduce our lab and my research named Identification of the Molecular Mechanism of Transcription Factors in Arabidopsis. First, I will explain the research in our lab and second, my research. Finally, I will talk about the experimental method used in my research. Now, let me introduce what kind of research we do in our lab. Our lab studies gene expression. Gene expression refers to the process by which DNA produces the final product protein. This picture is the central dogma. In simple terms, the process of making RNA from DNA is called transcription, and the process of making protein from RNA is called translation. Genes are units of function in DNA and encode proteins, which determines the function of cells. In this process, transcription regulation is the most important and most genetic regulation occurs. Gene regulation refers to controlling the amount of transcript or protein that is the product of a gene by controlling the expression of the gene in the process of DNA becoming a protein. Therefore, the expression level of each gene in the cell determines the function of the cell. So our lab focus on transcription factors. Why do we do transcription factor research? Let's take a closer look at the role of transcription factors. Transcription factors play a role in responding to the environment, regulating development and cell cycles, and responding to intercellular signals. As I said before, this is why transcription factors are very important for gene expression. Our lab mainly studies the transcription factors of Arabidopsis. First, we establish a network between family members of each transcription factor, and we work to identify molecular mechanisms for transcription factors. There are 6 types of transcription factors. Among them, I am researching the molecular mechanism of zinc finger protein related to the gravitational reaction of plants. Next, I will introduce my research. First, I will talk about tropisms. Tropisms refers to movement of a plant to bend in a specific direction by various stimuli as the plant grows. There are 3 types of tropisms in plants: phototropism, gravitropism, and hydrotropism. First, as you can see in the picture, phototropism refers to the phenomenon of plants growing in the direction of the sun. Next, hydrotropism is a reaction to the water. In the absence of water, the root of the plant grows straight, but as shown in the figure, when water is partially present, it refers to a phenomenon in which the root grows while bending in the direction of water. Finally, gravitropism is a phenomenon in which plants grow in response to gravity. Among these, I am currently working on gravitropism. Next, I will introduce about gravitropism. Inside of the plant stem, the outermost part is endodermal cells. They have a stateless capable of detecting gravity. When gravity is sensed, stateless accumulate downward. Then calcium proton and inositol 3-phosphate act as the second messenger to relocalize the oxygen transporter, spreading in various places in the columnar cell of the root, in the direction of gravity. After that, oxygen transport occurs. At this time, oxygen flows downward from the stem, and oxygen from the stem promotes the growth of the stem, so that the cell at the bottom of which oxygen is concentrated becomes large and bends upward against the gravity reaction. It will show negative gravitropism. On the other hand, in the root, since oxygen inhibits the growth of the root, the cell at the bottom becomes smaller and it shows a positive gravitropism that it bends downward in the same direction as gravity. Why should I study gravitropism? Because gravitropism is important for the plants to sustain life. Gravitropism helps the roots anchor to the soil and to get water. It also allows the stem to get sunlight for photosynthesis and oxygen for respiration. Plants have two forms. One is compact plant architecture and the other is spread out plant architecture. The compact plant architecture has the advantage of reducing damage to pests or pathogens, but also has the disadvantage for photosynthesis due to the small unit area for receiving sunlight. Also, spread out plant architecture is thought to be large enough to accept sunlight, but in fact it is inefficient because it creates a lot of shadows. It is also prone to damage from pests and pathogens easily. So I'm doing this research to make ideal plants for increasing plant productivity in all situations. The transcription factor that I'm currently studying is SGR5. SGR5 is one of the zinc finger proteins, and this is a major factor involved in the plant gravitropism. As you can see in the figure, if you look at the SGR5 mutant and wild-type core 0, you can see that the reaction speed of gravity is noticeably different over time. Such as if SGR5 does not function in plants, the gravity reaction is inhibited. Research continues to find out molecular mechanisms for target proteins that together affect these functions of SGR5. Next, I will describe the experimental method used in my research. Gateway cloning is the first step in the experiment. By the cloning, a gene is inserted into an expression vector according to the purpose. First of all, the product with ATTP1B2 attached to my gene by PCR is inserted into the CC division site of the donor vector with ATTP1 and P2 site. This process is called a BP reaction because it occurs at ATTP and ATTP site. After the PP reaction is complete, it becomes an entry vector with ATTL1 and L2 sites, and I can insert it into the expression vector I want to use. In the process of putting in the expression vector, the genes of the entry vector with ATTL1 and L2 sites are inserted into the CC division positions of destination vector with ATTR1 and R2 sites. This process is called error reaction because it occurs at ATTR and ATTR sites. After the error reaction is over, the expression vector is completed. Next, when cloning is finished, Is2 hybrid assay is performed. Is2 hybrid is a method for confirming protein-protein interaction and was used to perform library screening using a transcription factor library and to find a target protein that binds to my protein. The principle of Is2 hybrid is simple. If there is no binding between bait in the binding domain and prey in the activation domain, the reporter gene cannot be expressed. However, if BD and AD are combined indirectly, the reporter gene LOCG is expressed, which confirms that the two proteins are actually interacting in yeast. In our lab, PDES32 is mainly used as a BD vector. This vector contains the Galpor binding domain, synthesizes leucine and grows in an amino acid media. And PDES22 is used as AD vector. This vector has a Galpor activation domain which synthesizes tryptophan and it grows in an amino acid media. To briefly introduce the overall process of Is2 hybrid against BDSGR5, first, BDSGR5 inserted into yeast strain competent cells to wake BDSGR5 competent cells. Then, BDSGR5 and AD transcription library are incubated together and selected in the media. After picking colonies and growing them in a test tube for 2 days, yeast plasmid prep is performed. After that, send sequencing to analyze candidates which are target of my proteins. Cloning of the candidates finally proceeds to Is2 hybrid between PDSGR5 competent cell and AD candidates. Co-immunoprecipitation is a method to confirm binding between proteins in vivo. Co-immunoprecipitation is a technique for precipitating protein antigens from solutions using antibodies that specifically bind to specific proteins. This process is used to isolate and concentrate specific proteins from samples containing thousands of different proteins. In this experiment, I used magnetic A bead. First, add the HA tag to the bead and then add the extracted protein. Although there are several types of proteins in the protein, only the HA tag protein is specifically bound in the bead tagged with HA antibody. To briefly introduce co-IP process, put the magnetic bead in the E-tube at the very beginning. After that, put the H-tag antibody and attach it to the bead. And then add the extracted protein to the E-tube. When the bead and protein are incubated by rotator, the HA tag protein attaches to the bead together with the MIG-tag target protein that binds to it. The magnetic bead is magnetic, so if you hold the magnet, the bead will settle into the magnet. At this time, when washing buffer is added and removed, other proteins that do not bind to bead are released. After that, only the protein binds to the bead remains in the E-tube, and it undergoes an elution process that drops the binding of protein to the bead at high temperature, about 65 degrees. Then centrifuge to settle down, and after leaving only the bead, the protein is analyzed through Western blot. When Western blot is performed by sedimenting HASGR5 and MIG-tag target on this principle, the following result should be displayed. First, since input detects my protein contained in total protein, the result should appear like this. In addition, in co-IP, bands should appear from HASGR5 single and HASGR5 and MIG-tag target as detection results by HA antibody. And As a result of detection by MICTEC antibody, the band should not appear in the place where only MICTEC target was expressed, but the band should appear when expressed with HASCR5 and MICTEC target together. The last method is gravity response test. First, after growing the plant in a long day condition, the plant is grown for about 3 to 4 weeks, and the plant is laid in dark condition and the angle is measured to conduct an experiment. Take a picture every 30 minutes and measure the angles as shown in the following figure and draw this graph. This is all of my presentation and thank you for listening.